building block of protein. In nature, how many amino acids is there? Total 21. Okay. Among them, how many are for essential amino acids? 9. Acha. Non-essential, rest of them. Anyone tell me the name of essential amino acids? বলো, তোমাদের এটা মুখস্থ রাখতে হবে কিন্তু। Anyone can examiner can ask you in the exam. Please name the essential amino acids. This is important for bio exam. টোটাল 9টা আছে, সব বলতে হবে, সব মুখস্থ রাখতে হবে। Okay, let's start. So today we are going to cover this. First is very important: serine, alanine, threonine. For exam point of view, first is very important. You have to know this. Everyone should read from textbook. I am telling you some brief introduction about these three. Serine, alanine, threonine, most important. Other are not so important, but you have to know them. Starting from serine. What is serine? It is non-essential and glucogenic amino acid. It has aliphatic hydroxy amino acid. This is the structure of serine: C-double group and H3 group. There one OH group is there on alpha carbon. সবাই চলে এসছে বাইরে থেকে? সব आ गया बाहर से? हाँ, बुला सबको बुलाओ, जब कोई जब बुला के लाओ सबको। सब आ गया? अच्छा ठीक, ओके। So serine is aliphatic hydroxy amino acid. It is non-essential amino acid and it is a glucogenic amino acid. Sources: in your exam, if anyone asks you a type of question, just highlight these things. Serine, brief introduction. What is serine? There is a aliphatic hydroxy amino acid. Why? Because it has a OH group here. That is why it is called hydroxy amino acid. It is glucogenic amino acid and it is non-essential amino acid. This is brief introduction. You don't have to write to these things. Just 2 to 3 lines very important and is very important. If you write 2 or 3 lines, you may get full marks. If you deviate from the main topic, you may get 0 marks. Understand? Next is sources of serine. There is 3 major sources. First one is phosphoglycerate source. 3-phosphoglycerate. By 3 types of reaction: dehydrogenation, transamination and removal of phosphate group. It can form serine, like 3-phosphoglycerate with the help of enzyme NAD+ with enzyme dehydrogenase. NAD+ convert into NADH plus H+ and form 3-phosphohydroxypyruvate. And then 3-phosphohydroxypyruvate convert into phosphoserine by the enzyme phosphoserine transaminase. Here it requires glutamate, it convert into alpha ketoglutarate. And from phosphoserine with the help of H2O, it convert into serine plus phosphoric acid with the help of enzyme phosphoserine phosphatase. Okay, so this is the first source of serine from phosphoglycerate. You just write this thing, you get your full mark. Just this thing. Next source is complete. Okay. Everything written in textbook. Don't have to write anything. Okay. Next is from glycine metabolism. Serine can combine into glycine by the enzyme serine hydroxymethyl transferase. Here it requires tetrahydrofolate and it will combine into N5N10 methylene tetrahydrofolate and from glycine. Here CH2 group is removed and from glycine. By this reverse of the reaction, glycine can combine into serine. From glycine by reversal of serine hydroxymethyl transferase reaction. By this reversal of reaction, it can form into serine. Okay, this is second source. Third source is serine may be also be formed by transamination of hydroxypyruvate with alanine. Alanine, hydroxypyruvate combine into pyruvate plus serine. These are the three major sources of serine. Okay, the sources of serine, three types. First is phosphoglycerate by reaction, three-type reaction, dehydrogenation, transamination and removal of phosphate group. Second is reversal of glycine metabolism, glycine combined to serine. Next is serine may also be formed by transamination of hydroxypyruvate with alanine. Okay. Next, catabolism of serine. Okay. Deamination to pyruvate catalyzed by serine dehydratase. Here serine combined to amino acid by removal of H2O by serine dehydratase and form amino acid. From amino acid will combine to pyruvate. This is the catabolism of serine. Metabolic function of serine. You see, serine is very important. You may get short note or long question from serine. Read this very carefully. See, one carbon group, this is one carbon group. Serine donates one carbon group to the one carbon pool by the action of serine hydroxymethyl transferase enzyme. Serine hydroxymethyl transferase enzyme. One carbon group methylene tetrahydrofolate is removed from serine and glycine is formed. This is first metabolic function. Next is cysteine. Serine is used for the formation of cysteine. Serine plus homocysteine form cysteine plus homoserine. Third is alanine formation. Serine is converted to alanine by dehydration followed by transamination. This is third metabolic function of serine. First is donation of one carbon group, second is cysteine formation, third is alanine formation. Next, phospholipid formation. Serine is used for synthesis of phospholipid. What is phospholipid? Where is present? It is present in cell membrane. Okay, very important, phospholipid. It maintains the fluidity of membrane. Okay. Serine is used for synthesis of phospholipids such as phosphatidyl serine. Next is drugs. Development of drug. Serine analog are used as drugs and they inhibit nucleotide synthesis like azathioprine. This is an anti-cancer drug, azathioprine. And cycloserine is an anti-tuberculous drug. Okay, these are the function of serine. First is donation of one carbon groups, next is cysteine formation, next alanine formation, next is phospholipid formation, next is dark formation, dark development. Okay, these are the function of serine. Okay, next choline synthesis. Serine is decarboxylated to ethylamine by a pyridoxal phosphate dependent decarboxylase. Serine combined to ethylamine with help of serine decarboxylase. Here PLP is required and from ethylamine. What is important? Serine is a component of protein. In phosphoprotein, serine serves to esterify phosphate group like casein. Glycogen phosphorylase is activated by phosphorylation, while pyruvate kinase and phosphofructokinase 2 are activated by dephosphorylation. This covalent modification serves as a mechanism of regulation of enzyme activity. Okay. In glycoproteins, the carbohydrate group are usually attached to hydroxyl group of serine or threonine residue of the protein. Serine forms the active group of many enzymes like serine protease, like trypsin, coagulation factors. Next, this is the overview of serine metabolism. See, serine can form from alanine, from glycine, from phosphoglycerate. Serine can serve in protein as a component of protein, amino acid. From protein breakdown, you can form serine. From serine, you can form glycine. You can donate one carbon pool. You can form pyruvate, and pyruvate make a component to alanine and glucose. It can form ethylamine and formation choline. Choline is generate acetylcholine, which is a nerve terminal neurotransmitter. It can form phosphatidyl serine. It can combine to other phospholipids. It can serine can form sphingosine. It can form cysteine. It can form serinocysteine. Okay. So serine is very, very important. Let's move. Serine. So this is serine. Sources of serine. There are three sources. Catabolism of serine. Metabolic function of serine. Choline synthesis. Serine is component of protein. Overview. Next is selenocysteine. This is 21st amino acid, selenocysteine. It is seen in active site of following enzymes like thioreductin reductase enzyme, glutamate peroxidase enzyme, which scavenges peroxides. 5' deiodinase. They remove iodine from hydroxine to make iodothironine. And selenoprotein P, the glycoprotein seen in mammalian blood. Replacement of selenocysteine by cysteine will lead to decreased activity of these enzymes. Deficiency of some of the enzyme with antioxidant function may be related with atherosclerosis, and concentration falls in selenium deficiency. This is 21st amino acid, selenocysteine. Next important topic, alanine. It is also non-essential and glucogenic amino acid. Alanine can be formed by transamination of pyruvate. The enzyme is alanine aminotransferase. Pyruvate plus glutamate combined to alanine plus alpha ketoglutarate. This is the enzyme ALT. This reaction requires pyridoxal phosphate PLP. ALT is elevated in liver disease. In liver disease, ALT level also increased. Under condition of starvation, the glucose-alanine cycle is special metabolic significance. Alanine is quantitatively the most important amino acid taken up by liver from peripheral tissue, particularly from skeletal muscle. It forms a major participant in inter-organ transport of nitrogen. Okay, so alanine is non-essential and also it is glucogenic amino acid. It is important for liver muscle inter-organ transport of amino acid. Glucose-alanine cycle, you can come in your short note question. Please note it down. Glucose-alanine cycle. The glucose-alanine cycle operates between skeletal muscle and liver. The main purpose is to transport ammonia, because ammonia is highly toxic. It damages brain, especially nerve tissue. So to remove it from our body. In some disease like hepatic encephalopathy, have you heard the term hepatic encephalopathy? Anyone? In end stage of liver disease, The function of liver is to detoxify ammonia. In hepatic encephalopathy, the liver is already damaged, so liver cannot detoxify ammonia, and ammonia build up in the blood. This ammonia can go in brain and start dysfunction of brain, especially a patient with cirrhosis. He found this kind of symptom, hepatic encephalopathy. This is a dangerous condition. Okay. So we give lactulose to remove the extra amount of ammonia from gut to intestine. Hepatic encephalopathy. So ammonia is a highly toxic substance. We have to remove from our blood. So glucose ion cycle maintain this, remove this ammonia from blood. The carbon spectrum of alanine from pyruvate which channel to glucogenesis. The cycle assumes more important under condition of fasting and starvation, because in fasting and starvation, when muscle breakdown start, alanine can be formed. The nitrogen arising from oxidation of branch amino acid in skeletal muscle is also transferred back to liver via glucose alanine cycle indirectly through alpha ketoglutarate, glutamate, and then alanine and pyruvate by sequential transamination. So everyone showed it from your textbook, glucose alanine cycle. Okay. Next, the alanine cycle is a major energy providing pathway to tissue with an obligatory requirement for glucose. The major role of cycle under conditions of starvation is to transport nitrogen for ureogenesis in a non-toxic form and carbon spectrum for gluconeogenesis. Two molecules of alanine can form glucose plus urea. The Fourier cycle only serves to transport carbon spectrum as lactate. So these are something about alanine. So what is beta alanine? Here the amino group is attached to the beta carbon. It is formed during the catabolism of pyrimidine bases like cysteine and uracil. It mainly used for synthesis of coenzyme A. As the degradation of pathway, beta alanine is aminated to malonate semialdehyde, then malonyl CoA. Carnosine is a dipeptide formed by combination of beta alanine and histidine. Carnosine is a major constituent of muscle. It can activate myosin contraction. Hydrolysis of carnosine to beta alanine and histidine is done by carnosinase. In deficiency of this enzyme lead to carnosiuria. Carnosine is methylated to form anseline, which is a source of 1 carbon unit. In our general practice, we sometimes prescribe carnosine. Commercially available drug is there. It give the improve the strengthening of muscle contraction. So patient suffering from weak muscle tone, generous weakness, we sometimes prescribe carnosine. So this is the function of carnosine. Commercially available drug is there, like carnishirot 500 mg, which are available. We sometimes prescribe carnosine. Sometimes some pharma company they are making carnosine with vitamin B12, with vitamin E, like this. Okay. Next, so these are something about alanine. This is important. Glucose alanine cycle important because you may ask short note or part of long question in your exam. beta-alanine. Next is threonine. Threonine is it is essential amino acid and it is predominantly glucogenic. It has 2 S-center carbon. This is cycle threonine. It has 2 S-center carbon, 1, 2, and hence it has 4 dia-stereoisomers. So remember, in your carbohydrate classes, we discussed dia-isomerism, stereoisomerism. Remember, na? Bule mone ache to? Remember, everyone? Stereoisomerism, dia-isomerism. Okay. So it has 4 dia-isomers, namely D-threonine, L-threonine, L-threonine, D-L-threonine. Threonine does not directly undergo transamination but undergo deamination formation, forming alpha keto-butyric acid, reaction similar to serine. The enzyme required is threonine dehydratase. In turn, alpha keto-butyric acid can be oxidatively decarboxylated to propionyl CoA, then converted to succinyl CoA, which enter the gluconeogenesis pathway. Threonine may be cleaved by threonine aldolase to give rise to glycine plus acetaldehyde. The acetaldehyde is acted upon by aldehyde dehydrogenase to form acetate, which is activated to acetyl CoA by thiokinase. Here, threonine may be considered as partially ketogenic also. The Weiss group of threonine residue and protein serves to provide a site for phosphorylation, as in case of serine. This Weiss group also serves for combining carbohydrate residue to protein so as to make glycoprotein. This Weiss group. Okay. This small topic threonine. Just have to read this. Threonine. These are not so important, but still have to read. Sometime may ask in exam. Aspartic acid or aspartate. It is non-essential glucogenic amino acid. Aspartate of transamination give rise to oxaloacetate in the TCA cycle. Aspartate amino aspartate or ST refer to amino group of aspartate to alpha ketoglutarate to form oxaloacetate. Aspartate plus alpha oxaloacetate plus ketoglutarate form glutamate. ST is increase in hepatic disease and cardiac disease like myocardial infarction. AST. Malate aspartate shuttle transfer the cytoplasmic NADH into mitochondria for oxidation in the electron transport chain. It is also used for transport of oxaloacetate into cytoplasm. Aspartic acid is an important member of urea cycle. The alpha amino group is directly contributed to the urea molecule. The carbon cycle of aspartate can also enter the glucogenic pathway as fumarate. Citrullin plus aspartate combine to arginine plus fumarates. Aspartate is for synthesis of purine, like fast is fast nitrogen and 6 NH2 group. The whole molecule of aspartate is incorporated into pyrimidine DA. The fast nitrogen and carbon 4, 5, 6. If you see the structure, you will find this. You read this when you are pyrimidine synthesis. You have to read this. So this is aspartate or aspartic acid. Next is asparagine. The name is originated from a fungus called asparagus. Aspartate react with ammonia to form asparagine. Asparagine can be hydrolyzed to aspartate and ammonia by asparaginase. L-asparaginase is an anti-cancer drug against leukemia and lymphoma. Because of these cells cannot synthesize asparagine. So, the enzyme will destroy the available asparagine in the blood. So, cancer cell will die. This is the use of asparagine. And it is a glucogenic amino acid. Okay. So, this is the use of asparagine. Use as cancer therapy. Glutamic acid or glutamate. It plays central role in metabolism of amino acid generated during transamination reaction. In transamination reaction, most amino acids transfer their amino group to alpha ketoglutarate acid to form glutamic acid. Example: amino acid plus alpha ketoglutarate amino transferase alpha keto acid plus glutamate. Glutamic acid is also formed during the metabolism of histidine, choline and arginine. Next is oxidative deamination. Glutamic acid is deaminated to form alpha ketoglutarate by the enzyme glutamate dehydrogenase with the help of NAD. Next is metabolism. Glutamic acid enters TC cycle as alpha ketoglutarate, gets converted to oxaloacetate to enter glucogenic pathway. Next is glutamine. It is synthesized from glutamic acid. Gamma carboxy glutamic acid is present in prothrombin. The gamma carboxyl group is added as the post-translational modification which needs vitamin K. Next is auxiliary neurotransmitter. Glutamate plays a vital role in the cognitive process involving the cerebral cortex, including thinking, the formation of memory and recall, and is vital for perception. The large neurons in the cerebral cortex use glutamate as their neurotransmitter. Okay. Glutamate is present in neuron. It serves as excitatory neurotransmitter. Next is glutathione. Glutamate is a constituent of tripeptide glutathione. It is used by bacteria for folic acid synthesis. Glutamic acid is decarboxylated by 2-GABA. What is GABA? Gamma aminobutyric acid. Glutamic acid on decarboxylation gives rise to GABA. Part of the glutamate in the brain can be shunted through GABA pathway and catabolized to succinate. GABA is an inhibitory neurotransmitter and it opens the chloride channel in post-synaptic membrane in CNS. So glutamate is excitatory neurotransmitter and GABA is inhibitory neurotransmitter. You may read it in your physiology classes. Okay. Glutamate and GABA. Next is glutamine. It is glucogenic amino acid synthesized from glutamic acid. The amidation of glutamic acid to glutamine is catalyzed by glutamine synthetase. Glutamic acid can react with a molecule of ammonia in presence of ATP. This reaction is important in ammonia trapping in brain as well as for transport of ammonia in a non-toxic form. You may read it in your urea cycle classes in details. This thing. So just listen this, read this. It is hydrolyzed to glutamate and ammonia by the enzyme glutaminase. The reaction is seen in the neural tube cells. This ammonia reacts with asparagine to buffer acids excreted in urine. The major feat of glutamine is to hydrolyze to glutamate and ammonia. Glutamic acid is then deaminated to alpha ketoglutarate and enters TC cycle for further catabolism. Other important functions of glutamate are the N atom 3 and 9 of pyrimidine are derived from glutamate. Third nitrogen of pyrimidine derived from glutamine. It is the source of additive group of guanine and cytosine, used as a conjugation agent, for example production of phenyl acetyl glutamine. It dominates the amino group of amino sugar and amide group of nicotinamide. So these are the very small topic today. Let's read these amino acids. First is very important: serine, alanine, threonine. So I put one. This is serine. Next is sources of serine. This source. Catabolism of serine. Metabolic function of serine. Next, choline synthesis. Serine serves as a component of protein. Overview of serine metabolism. This is very important in serine. Okay. Next, cellular cysteine. This is the 21st amino acid I put here because this is a small discussion today. Next is alanine, second important topic, glucosylation cycle, beta-alanine, threonine, aspartic acid, asparagine, glutamic acid, glutamine. Thank you.